Okay, so this is just gonna be a little bit different. I'm on the road. I'm gonna be using my laptop instead of my typical setup, but we'll get to this. So we left off last time talking about fatty acid oxidation. So this example that he gives is just sort of a general example. It is not the specific one necessarily that you need to know. So this is how it's going to work with all of our different fatty acids. This is going to uses the example of palmitoyl CoA, but understand that this applies to all sorts of different fatty acids. Our main focus, we want to focus on the enzymes here. That will be our main focus for this. We'll go ahead and look at the steps. So the first step within the fatty acid oxidation is the beta oxidation of the beta carbon of the fatty acid, followed by a hydration, an oxidation, and a thiolysis. In this manner, a series of reactions occur with successive removal of 2 carbon fragments as 1 molecule of acetyl-CoA. So when we're looking at our fatty acid chain, we have an alpha carbon and we have a beta carbon. So this whole process is going to be occurring right here between that alpha and the beta, so at that beta carbon, and we'll break off a 2 carbon molecule that will be acetyl-CoA when it breaks off, and this process will then continue down. So we'll have a dehydrogenase, a hydratase reaction, a dehydrogenase, and then that thiolase reaction, eventually giving us an acetyl-CoA to come off that can go off into the Krebs cycle. And this whole process will just repeat over and over and over and over again until we've split all of our 2 carbons up off and made as many acetyl-CoA as we possibly can. So the oxidative conversion of 2 carbon units into acetyl-CoA from the carboxyl end of the fatty acyl chain with simultaneous production of NADH. We have oxidation of acetyl-CoA into CO2 via the citric acid cycle with simultaneous generation of NADH and FADH2. So this is talking about how that acetyl-CoA is now then going to go into that citric acid cycle from being broken down off of the fatty chain. So then the production of ATP from NADH and FADH through the respiratory chain. This is now talking about the electron transport chain all through there. Sorry, my mouse is a little bit weird. So this is just kind of listing out the next steps, kind of reviewing what we talked about in Try 1. So this provides energy for ATP biosynthesis through oxidative phosphorylation. So if we consider a fatty acid as the palmitoyl CoA, this is just the example now, it is a 16 carbon fatty acid, then there would be a total of 7 cycles to remove a total of 8 molecules of acetyl-CoA. The 8th acetyl-CoA arises from the very last cleavage. So we can watch this whole process. So the palmitoyl CoA, right, has everything on here. So our acyl-CoA dehydrogenase is going to actually act on this bond right here between the beta and the alpha carbon. What it's going to do, it's a dehydrogenase, so we are removing hydrogens, and by removing hydrogens, we change this bond structure. See? So the bond structure here is going to change. We then have a hydrolase where water comes back in. We then are going to have another dehydrogenase reaction. Again, this is all of this. You can look at these 3 steps: the dehydrogenase, the hydratase, and then the second dehydrogenase. These are kind of our setups to kind of to prepare the area that we're going to break off. So you can see how this changes how we have these CH2, CH2, how it changes now to a C double bond C, and this H, and then we have an O over here as well. And then the H2O comes in, and how again we change its structure until finally the dehydrogenase, we have it where we want to be. And now we have the acyltransferase, or the thiolase, which is going to bring in another coenzyme ASH and plant it right there and finish cleaving this off to give us our acetyl-CoA. So I'd go through, look specifically at the enzymes. The enzymes are what we're going to want to know. That's what's most important, okay? And then the steps of them. So it's a dehydrogenase, then a hydratase, a dehydrogenase, and then a thiolase. Then this whole process now, so originally we started off with a palmitoyl-CoA, and now we have a myristoyl-CoA. This is just saying how many the number of carbons that there are. So we cleaved off 2, and now from 16, now we're at 14, and this whole process repeats with the same enzymes over and over and over again, cleaving off and cutting off carbons until finally we get down to our last 4. And what's special about our last 4 is we actually, the very, very last reaction, we get 2 acetyl-CoA because it's going to split it, and then we only have 4 total, so it's going to split it into 2 carbon acetyl-CoAs. So that's why when we talk about down here, we talked about a total of 7 cycles to remove 8 molecules of acetyl-CoA. You can kind of look at this as almost like a formula where we take the total number of carbons in the fatty acid, divide that by 2, and then subtract 1 is going to be our total number of cycles. You'll want to remember this. You'll have a couple of questions literally specifically just asking this: how many cycles will it take for this specific fatty acid, or a fatty acid with this much that has this much many carbons in its chain. So just remember to keep that in mind. So moving on, we have beta oxidation of saturated fatty acids, sequential steps. So we have 4 enzyme catalyzed reaction steps. Removes 1 acetyl-CoA at a time from the carboxyl end of the fatty acyl chain. Just going to throw this out here as well. We talk about different types of fats. We have monounsaturated, polyunsaturated, saturated, and then trans fats, right? What this is really saying, it's talking about actually how the bonds are structured and the hydrogens that are on that fatty acid chain. So if we look up here, see how CH2, 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 if every single one of these had a CH2, so in other words they're full of hydrogens, that is going to be a saturated fat. When we get less of them, so for example, if we have, say we have carbon, let's say we just have a 3 chain right here. This one only has 1, so it forms a double bond. This is a monounsaturated fat. Let's say this one only had 1 hydrogen. Now it is a polyunsaturated fat. When every single carbon has its total hydrogens, actually there would be a 3rd hydrogen like that. Now that is a saturated fat. So it's talking about the different structures of how the bonds are and how saturated it is with hydrogens. And that's why when you talk about saturated fats being worse for you, it's because they're actually more difficult to break down for the body to use as energy. Whereas our mono and poly unsaturated fats, if we look up here at the breaking down process, see how we had to insert a double bond into them in order to do this process? If we have an unsaturated fat, there's already going to be double bonds, which makes it just easier to break down and actually use that specific fat. You'll talk more about all of that in your clinical nutrition class in a couple of tries, but it's good to think about it right now. Okay, so continuing and moving on. So 4 enzymes of each pass to remove 1 acetyl-CoA at a time are, these are just going to review, this is all just going to be a review of those specific enzymes and kind of what they do, going into a little bit more describing what they actually do. So the acyl-CoA dehydrogenase is our first one. And then we have our enoyl-CoA hydratase, our beta-hydroxyacyl-CoA dehydrogenase, and then our acyl-CoA acetyltransferase, aka our thiolase. So reaction step 1. Dehydrogenation of alkane to alkene. So we talked quite a bit, it came up a couple of times, talking about alkane to alkene last try. Hopefully you remember these words a little bit. Hopefully they're a little bit familiar. So the acyl-CoA dehydrogenase catalyzes the formation of trans 2,3 double bond and produces trans delta 2 enoyl-CoA. The enzyme bound FAD is reduced to FADH. So there's a lot in here. I'm going to try and point out like the specific things that you're going to want to know. So you want to know the name of the enzyme, the acyl-CoA dehydrogenase. And if we think back at dehydrogenase, what is it actually doing? What it's doing is removing hydrogens. So if we have a carbon structure and we're removing hydrogens, what's going to happen is that double bonds are going to take place. And that's all that this is saying, this trans 2,3 double bond. It's just saying that between the 2 and the 3 carbon, you're getting now a double bond. And in that process, you also get the formation of FADH2. Okay? Next step. So now we've gone alkane to alkene. Now with a hydration reaction, we're going from an alkene to an alcohol. And these, what this is talking about, these specific names, is just referring to the example that's above. But understand again that this can happen like to all of them. Okay, so don't get too locked up in these names. So the molecule, the fatty acid chain, is then hydrated by the addition of water. And then the double bond is now saturated to form L-beta-hydroxyacyl-CoA. And that reaction is catalyzed by the delta 2 enoyl-CoA hydratase, aka crotonase. So again, we'll want to remember our enzyme. And then we'll want to remember that it's a hydration reaction. And that means that the double bond is then saturated. So just double bond getting saturated means that now you're getting rid of the double bond and adding more hydrogens back into it. Reaction step 3. Dehydrogenation of alcohol to a keto compound. So the L-beta-hydroxyacyl-CoA dehydrogenase, this is our next enzyme, catalyzes the next oxidative reaction to form beta-ketoacyl-CoA with simultaneous production of NADH from NAD+. Apologize, I'm still getting over a little bit of a cold. Okay, so that L-beta hydroxyacyl-CoA dehydrogenase catalyzes the next oxidative reaction to form that beta ketoacyl-CoA, and then we get NADH from NAD+. Again, remember the enzyme name. Remember what it does. So it's going to catalyze the next oxidative step. So we get NADH now. So in the very beginning, we get FADH2. After that, then we're gonna break away. We're gonna form the double bond, and then, or we're gonna. Sorry, I apologize. We're going to form the double bond in the beginning and make FADH2. Then we're going to get rid of the double bond. We're going to resaturate it, and then we're going to get NADH. Now the final step is the thiolase. So it's the transfer of the fatty acid chain and the release of the acetyl-CoA, which is the whole goal of this process. So the fourth and the last reaction step in the beta oxidation of fatty acid cycle is thiolysis of the C2C3 bond. Remember, the C2 is our alpha carbon and the C3 is our beta carbon. So it's that bond which releases acetyl-CoA and produces new acyl-CoA with 2 carbons shorter than the starting fatty acid substrate. This reaction is catalyzed by acyl-CoA transferase, also known as thiolase. So this sequence of reactions is repeated until the acyl-CoA has been converted completely to acetyl-CoA. For example, for the oxidation of palmitoyl-CoA, the sequence takes place 7 times to finally produce 8 acetyl-CoA. The oxidation of fatty acids are highly exergonic. If you remember, we talked about exergonic. That exergonic is referring to the energy. So we're going to be releasing a lot of energy, so that's going to produce heat. So all the acetyl-CoA generated through the oxidation palmitoyl-CoA enters the citric acid cycle. The FADH and NADH produced produced by beta oxidation and the citric acid cycle can be reoxidized by the electron transport chain, and this ultimately generates a total of 129 ATP molecules, and this is all just from one fatty acid chain. So we can see how much more energy that there really is in fats than compared to just glucose. And that's actually why it's so much more difficult to lose fat than it is to just burn glucose, because with fat you have to use a lot more energy to burn fat. Okay, there's our overall reaction. So we see the using the CoA, the FAD, NAD+, and H2O, and we get that FADH2, the NADH, and a couple of extra hydrogens as well. And then we get 8 acetyl-CoA from a 16-chain palmitoyl-CoA. So fatty acid catabolism for energy. Each of those cycles generates 1 mole of the FADH2, so we get 1.5 moles of ATP. We get 1 mole of NADH, which is 2.5 moles of ATP, which is the same as before. Hopefully you remember that. And the acetyl-CoA enters the citric acid cycle and is further oxidized into CO2. And this is going to make more GTP, NADH, and FADH2. So the yield of ATP during oxidation of 1 molecule of palmitoyl-CoA to CO2 and H2O is as follows. You can see all of the different processes from the Krebs cycle and how much FADH2 and NADH and GTP we're going to be producing, which gives us then those totals of ATP being produced. So it's important, though, to remember not every single fatty acid chain is going to have an even number. When it doesn't have an even number, when there's an odd number, the very last cleavage, we're going to produce an acetyl-CoA and then also a propionyl-CoA from that last cleavage, propionyl-CoA. If you think back, hopefully you remember this. Propionyl-CoA is one of the intermediates that we talked about. I don't remember exactly in which process, but in one of our processes of converting everything back into the Krebs cycle, propionyl-CoA was one of the intermediates. And so that propionyl-CoA can be converted to succinyl-CoA, which can then enter the citric acid cycle or the Krebs cycle through that path. So the oxidation of unsaturated fatty acids requires additional enzymatic steps. So we can see here, okay, we'll come back to the picture itself. So enyl-CoA isomerase, the monounsaturated, is required to convert the cis isomer to the trans isomer. And then we have NADPH dependent 2,4-dienyl-CoA reductase. Polyunsaturated is required to saturate the double bond into the unsaturated portion. There's our isomerase right there. Kind of a random spot to throw this in here. If I'm not mistaken, we'll talk more about this later on as well. Alpha oxidation of fatty acids. This is a specialized pathway in the brain. It is a removal of one carbon at a time from the carboxyl end of the fatty acid. This has been found in the brain tissue. We do not need the CoA intermediates, and this does not generate high-energy phosphates. Then we also have omega oxidation of fatty acids. This occurs in the microsomes involving cytochrome P450 enzyme system, and it produces dicarboxylic acid. If you remember the P450 enzyme system, this was dealing with toxins like alcohol in the liver. We also have paroxysmal fatty acid oxidation. This differs from mitochondrial oxidation. We have very long-chain fatty acids from a high-fat diet are oxidized in the peroxisomes. There's no carnitine needed, and these will produce less energy. Don't worry too much about these. I would understand that they exist. And know the general information. It's like the alpha happens in the brain. It's one carbon, and there's just no high-energy phosphates. The omega is involved with the P450 enzyme system. That's pretty much all you should know. You need to know about that one. And the paroxysmal is for very long-chain fatty acids, and it produces less energy. Okay, now getting into ketone bodies. Hopefully, a lot of people have heard about keto before. Ketone bodies are what are going to be produced when we're just breaking down the fats. So the synthesis of ketone bodies occurs in the liver mitochondria when fatty acid concentrations are high in the blood. So during fasting, starvation, or untreated diabetes, or a high-fat diet. Any of those can be possible reasons that you'll see ketone bodies. So there's two fates of acetyl-CoA produced during beta oxidation of fatty acids. The acetyl-CoA can enter the citric acid cycle or go into ketogenesis, which is an alternative pathway. In mammals, it undergoes conversion to acetone, acetoacetates, and beta-hydroxybutyrate. These three are our ketone bodies. We'll look at our step-by-step of the ketogenesis. I want to make sure that we have, because this one specifically is showing the acetoacetate formation. So acetoacetate is formed when two acetyl-CoA units condense. Beta-hydroxy beta-methylglutaryl-CoA formation, or HMG-CoA. You'll see that one a lot more, HMG-CoA. Formation is catalyzed by hydroxymethylglutaryl-CoA synthase. In this reaction, one molecule of acetoacetyl-CoA condenses with a third molecule of acetyl-CoA. D-beta-hydroxybutyrate formation is catalyzed by beta-hydroxybutyrate dehydrogenase. In this reaction, acetoacetate is reduced in NADH-dependent reaction. The HMG-CoA lyase cleaves HMG-CoA to form acetyl-CoA and acetoacetate. And then the acetoacetate can be spontaneously decarboxylated to produce acetone. Okay. So looking at all of these, we have two acetyl-CoA molecules. A thiolase essentially is going to smush these two together, giving us acetoacetyl-CoA. Another enzyme, HMG-CoA synthase, puts in another acetyl-CoA and gives us our beta-hydroxy beta-methylglutaryl-CoA. Then we have an HMG-CoA lyase that brings us to acetoacetate. The acetoacetate can follow one of two pathways. It can go down with a dehydrogenase to form hydroxybutyrate down here, and also using NADH, or it can get turned into acetone. Not a ton here. I would focus more, make sure that you know what the ketone bodies are, and then just briefly a little bit about the steps. Okay, look at the enzymes, think about the process, the overall process, where we have two acetyl-CoAs. They get smushed together to give us our acetoacetyl-CoA, and then they get continuously broken down by the HMG synthase and the HMG lyase to give us our acetoacetate, which is one of our ketone bodies. And the acetoacetate can become either of our other two ketone bodies, acetone or the hydroxybutyrate. Looking at the condition at which ketone bodies are generated. So normally, acetyl-CoA will enter the citric acid cycle through its combination with oxaloacetate and is used for gluconeogenesis. So if there is a high demand for glucose, but the oxaloacetate levels are low, meaning that there isn't enough oxaloacetate for it to combine with, the acetyl-CoA does not enter the citric acid cycle. Instead, it is converted to ketone bodies in the liver. The ketone bodies are then transported in the blood circulation and arrive in tissues such as the muscle and the brain, especially the brain. Muscle and brain tissue converts the ketone bodies to acetyl-CoA, which is then oxidized via the citric acid cycle. Thus, muscle and brain tissue can generate energy when there is a short supply of glucose, because these are our most important parts of our body. Specifically, think about what other muscles we have. Think about like the heart. The heart is a muscle. And then the brain, we need the brain to do everything that we do every single day, all the time. So those are going to be the main focus. So when there's high demand for glucose, but oxaloacetate levels are low. Ketone bodies are overproduced in situations like diabetes. So insufficiency of insulin. So you don't have enough. Either you don't have enough insulin, such as in type 1 diabetes, or the cells are not as sensitive to insulin, like in type 2 diabetes. You get a decreased glucose uptake by the extrahepatic tissue. As a result, malonyl-CoA, which is the starting material required for fatty acid synthesis, levels are very low. This leads to activation of carnitine acyltransferase 1, which transfers fatty acyl-CoA into the mitochondria, followed by their degradation to acetyl-CoA. However, acetyl-CoA cannot pass through the citric acid cycle since the intermediates of citric acid cycles are withdrawn to use them as the substrates for gluconeogenesis. So specifically, no diabetes, then the rough why, it's because of the insulin, and then you can't get the glucose into the cells. And then also starvation, where gluconeogenesis depletes citric acid cycle intermediates. The overproduction of ketone bodies can lead to acidosis or ketosis. Know these two. You'll sometimes talk about, like, with ketosis especially, it'll be pretty interesting. The patient's breath actually has a fruity smell, like a fruity or a sweet smell to it. That's actually how they can tell if a person is in ketosis. Also works the same with animals. Just a little clinical correlates. So type 1 diabetes is caused by insulin deficiency due to autoimmune mediated destruction of insulin producing cells in the pancreas. Do you remember what those cells are? We have our beta cells. Our beta cells are insulin producing cells. So deficiency of insulin causes diabetic ketoacidosis and results in severely high blood levels of glucose and increased synthesis of ketone bodies. Because you think we need our insulin to bind to its receptors in order to allow glucose through. So even though our blood glucose levels are really, really high, we have plenty of available glucose, we can't get it into the cells because of not having enough insulin, or actually having no insulin in the case of type 1 diabetes. And then we get the production of ketone bodies that can travel and cross over through the boundaries of the brain and the muscle to keep us alive. Okay, and that is the end of chapter 1. Thank you for joining me. Remember to study hard. Go back through this a couple of times. Really set up this foundation of understanding kind of the beta oxidation, understanding the fats, to set yourself up a little bit better for what is to come in the next couple of chapters.